y ya están tomando asiento. Nada más quiero dar un anuncio rapidísimo antes de que continuemos con las pláticas. Les recuerdo que es la fiesta de bienvenida de los nuevos ingresos. El evento empieza a las 9. Ustedes ya sabrán que es un evento de pieza cultural. Va a estar muy padre, es una obra de teatro que se llama Tandas de humor y faltas. Es un show medio cabaretesco, muy padre, pero las entradas son limitadas, entonces quien va a llegar tiene que llegar puntual. Vamos a estar repartiendo los pases a partir de las 8:30 en el Tierra Luna, con preferencia a los nuevos ingresos, por supuesto, pero tienen que ir con credencial de Bilingual para que les demos los pases y que no paguen cover. Quien no llegue con credencial o quien no sea del instituto puede pagar un cover de 120 pesos en el lugar, así que yo creo que es muy padre que aprovechen, lleguen al evento y que escojan mesa de una vez. Y pasen la voz porque creo que faltan muchos ya, pero los esperamos hoy. Y ya saben que después de la hora de teatro, que también yo digo que va a estar súper padre. Viene la fiesta, así que los que no quieran llegar al evento cultural, pueden llegar después al reventón. Vale, gracias.
Bueno, pues continuando con el programa, vamos a dar la bienvenida al doctor Christopher Irwin Smith. En el laboratorio del doctor Smith se utilizan técnicas de biología molecular y genética de poblaciones para responder preguntas aplicadas a ecología y evolución. Sus áreas de interés incluyen la coevolución de las interacciones planta-insecto, la formación de especies y la radiación adaptativa. Actualmente laboran en la Universidad de Wyoming, en el estado de Oregon. Sus publicaciones suman más de 500 citas. Démosle la bienvenida con un aplauso.
Buenos días. Al empezar quería agradecer a los estudiantes por la invitación y por hospedarme aquí. Esta visita ha sido un periodo padrísimo. Y también quiero agradecer a todos ustedes por su atención y también quiero pedirles perdón porque otra vez va a ser una plática en inglés. I'm going to tell you today about work that my lab has been doing over the last 10 years, looking at the role of coevolution in species formation, particularly within ovulative pollination mutualisms. And I was very struck by there it is. I was very struck by some of the talks this morning. Think about the connection between historical biogeography, ecological biogeography, and population genetics. In that, I initially started this work being primarily interested in historical biogeography and the history of the North American deserts. But increasingly, my focus has moved towards really ecological biogeography and population genetics. And this shift over time, I think, comes from sort of a fundamental problem in evolutionary biology, which is that as evolutionary biologists, we often seek to understand evolutionary processes by examining evolutionary patterns. However, frequently this is difficult because multiple distinct evolutionary processes can often give rise to very similar evolutionary patterns. And so it's often difficult to distinguish which particular evolutionary process gave rise to the pattern that we're observing. And in my case, I'm interested in trying to understand the role of coevolution. And historically, I've looked at evolutionary patterns that seem to be diagnostic of coevolution. However, work that has developed over the last 10 years or so tells us that frequently extrinsic processes, including selection to the abiotic environment and biogeographic history, can give rise to patterns that are the same as those that we would expect under coevolution. And so just to outline this morning's talk, I'm first going to make an argument to you that frequently the specific patterns that we might expect under a coevolutionary scenario are very similar to those that we might expect due to similar extrinsic biogeographic history. I'm then going to tell you some about the natural history of the Yucca system, with a particular focus on the relationship between Joshua trees and their pollinators. I'll then tell you about some somewhat old work now that I've done looking at the phylogeography of Joshua trees and trying to distinguish phylogeographic processes from evolutionary processes. And then for the end of the hour, I'm going to tell you about some newer work that we're using both experimental and population genomic approaches to try to identify signatures of reciprocal selection and reciprocal adaptation. So to begin, one of the foremost questions that evolutionary biologists seek to address is the diversity of life on Earth, and in particular, what explains relative disparities in diversity between different groups of organisms. So for example, the flowering plants is one of the most spectacularly diverse group of organisms. Plants in general account for about a quarter of all described terrestrial eukaryotes. And within the terrestrial plants, the flowering plants are far and away the most diverse, accounting for about 90% of all described species. There are about 200,000 species of flowering plants. And this spectacular diversity is particularly surprising given their relative age. Some of the oldest fossil angiosperms date to about 135 million years ago, whereas groups like the mosses and liverworts have a fossil record beginning about 400 million years ago. And yet this group is much less speciose and much less diverse. And famously, Charles Darwin described the diversity of flowering plants as an abominable mystery. In this letter to J.D. Hooker, he talks about the diversification of flowering plants as potentially being a problem for evolution or a problem for a gradualist model of evolutionary change, because Darwin couldn't understand how such spectacular diversity could have arisen so quickly. He actually expected that perhaps there was somewhere a lost continent under the sea where we might find the fossil history of the angiosperms. And in the same letter, he remarks that a French botanist, Porto, had made some speculative arguments that perhaps the diversity of flowering plants might be somehow connected to pollination by specialized insects. And this idea received some support about a century later from Vern Grant, the famous 20th century botanist, writing in one of the first issues of the journal Evolution. Grant simply looked at the characteristics that botanists may use to try to distinguish closely related species, say species within the same genus. And what he noted is that when we look at groups of plants that tend to be pollinated by specialists, so groups of plants that are associated with bird or bee or bat pollination, those groups of plants tend to be differentiated most, or botanists tend to use features of the floral morphology to distinguish between species. Whereas when we look at groups of plants that are either generalist pollinated or rely on abiotic mechanisms such as wind or water, those groups of plants tend to be distinguished more easily based upon vegetative features than on floral features. And simply on the basis of these differences, Grant argued that perhaps pollinator-mediated selection, or selection that's induced by pollinating insects, may actually be a major force driving speciation and the diversification of flowering plants. And there are a number of macroevolutionary patterns that we can see today that lend further support to this idea. I'm just going to talk about two of them. And one pattern that we observe very widely across plant-pollinator interactions are these patterns of phenotype matching, where a feature of a pollinator matches some corresponding feature of the plant. Famously, we can see this in the case of Darwin's orchid, which has this enormously long 13-centimeter nectar spur, which we now know is pollinated by this hawk moth, Xanthophaeum morgani predicta, which also has a proboscis 13 centimeters long, or just long enough to reach the nectar down the bottom of this nectar tube. Or to look at some more recent work, these are data from figs and fig wasps. This is from George Weibel's lab, in which he looked at variation in styling across different species of fig flower, and we see that there's a very strong correlation between the length of the style and the length of the fig wasp's ovipositor, which is going to be used to layer eggs inside the flower. However, Scott Newsmer has argued rather strongly that these patterns of phenotype matching are not necessarily indicative of a history of reciprocal adaptation and co-evolution. Indeed, a number of scenarios that do not involve strict sense coevolution can give rise to phenotype matching. And through both modeling and simulation work, Scott has shown that in fact we can generate patterns of phenotype matching even in the absence of any biotic selection. So even when there's no selective interaction between this insect and the plant, we can still generate patterns of phenotype matching simply due to variation in the abiotic environment. And so I've drawn sort of a cartoon here, because when I asked Scott for the data, he sent me some equations. And so we might imagine that this is a landscape that varies in both temperature and rainfall, and selection in this abiotic environment might, for example, favor larger body size in our moths and larger corolla in our plants. And you can argue about whether or not that's a realistic idea, but in the simulation, we see that this can arise even without coevolution. Another pattern that we commonly associate with very strict specialized coevolutionary interactions are patterns of phylogenetic congruence, or that is when we build phylogenies of, say, a pollinator, we often see sort of mirror image phylogenetic relationships in the plants that are associated with, perhaps suggesting a long history of mutual association and co-speciation. However, throughout this talk, we've seen how we can generate common phylogenetic or phylogeographic patterns across distantly related taxa that may not be interacting with one another strongly. Most famously, of course, in the case of Gondwana biogeography, where we can see similar phylogenetic patterns across distantly related groups of organisms. So with that context now, I'd like to focus in on thinking about the relationship between yuccas and yucca moths and work that my lab has been doing with the Joshua trees. So yuccas and yucca moths are, of course, a textbook example of an obligate pollination mutualism in which both the plant and the pollinator are entirely dependent upon one another for reproduction. Yuccas are pollinated exclusively by yucca moths, and yucca moths can reproduce solely by laying their eggs in a yucca flower. Typically, although not always, there's also a strict one-to-one specificity in which each host species of yucca is pollinated by a single species of pollinator. In addition to this remarkable specificity, yucca moths exhibit a highly unusual and very stereotyped pollination behavior. I'm going to try to show a video of this. We'll see if it works. Major bummer. Hold on a second. Okay. Technology. Okay, so what we're seeing here is we're looking down into the inside of a flower, inside of a yucca flower. I apologize for the... Okay, I apologize if the quality is a little bad here. This is a video made by an elderly Swedish gentleman. So, but we're looking down into a yucca flower. You can see the yucca moth down at the base. And in a minute, what's going to happen is that she's going to move up and crawl up to the top of the pistil, or the top of the female part of the flower. Come on. Come on. Okay, here she goes. And you see her doing that kind of pecking motion. What she's doing is she's actually depositing pollen directly onto the stigma or directly on the receptive part of the flower using a set of uniquely derived tentacle-like mouth parts. These tentacles are actually not homologous to any other insect mouth parts. And here you can see this same behavior in Joshua tree. And the reason that she's willing to use some teleological language to engage in this very elaborate pollination behavior is that immediately before pollination, she's going to deposit some eggs inside the flower. Those eggs are then going to hatch and develop into larvae or caterpillars that will consume a portion of the seed crop. And because the yucca moth is the only pollinator, the only way to ensure that there's food available for the offspring is to ensure that the plant is pollinated. And so we could imagine, using again some sort of teleological language, that In exchange for very reliable pollination, the yucca is willing to sacrifice some of its seeds. And on the other hand, in exchange for getting a very nutritious food source for its offspring, yucca moths are willing to go to the effort of pollination and with considerable risk of moving pollen between different plants. So focusing in now on one particular group within this association, I've been looking at relationships of the Joshua tree. Here are some photos of it. Joshua trees are endemic to the Mojave Desert in southwestern North America. And they occur in this series of sort of high elevation, fragmented range patches around the periphery of the Mojave Desert. Their range is bisected by a series of deep, very low elevation, extremely arid, very hot desert environments, including the Atacosa Desert, Death Valley, and the Salton Trough. And historically, botanists have noticed that Joshua trees occurring on either side of this divide look morphologically different from one another. So Joshua trees occurring west of Death Valley tend to be taller and more tree-like. They tend to have more branches and longer leaves, whereas Joshua trees occurring east of Death Valley tend to be shorter and more bush-like with more branches and shorter leaves. In addition, work that was completed by my postdoctoral advisor, Philip Lemire, showed that these two different morphotypes are actually associated with two sister species of yucca moth, Tegatecula synthetica, occurring in the west, and Tegatecula antithetica, occurring in the east. And the distribution of these two moths, pollinator species, coincides exactly with the distribution of these two morphotypes. And on the basis of these morphological differences and the differences in pollinators, botanists have recommended that we actually split Joshua tree into two separate species, two sister species, Yucca brevifolia, occurring in the west, and Yucca jageriana, occurring in the east. And to help you sort of keep track of these various layers throughout this talk, I'm always going to present data from Yucca brevifolia or Tegatecula synthetica in shades of blue on the left hand or the western side of the screen, and I'm also going to present data from Yucca jageriana in shades of green on the right hand or the eastern side of the screen. Okay. In addition to these differences in growth morphology, within the Joshua tree system, we also see patterns of phenotype matching, similar to what I showed you earlier for figs and fig wasps. That is, we look at the flowers too, that looks really dark. Okay, so these are false colored, so these are stained Joshua tree flowers that have been bisected. And you're supposed to be able to see right here in white are the ovules, or the part of the flower that become the seeds. And this spot right here is where the female yucca moth is going to deposit her eggs during oviposition. So that's where her babies need to go. This distance here, where the ovules are up to the stigma, we define that as the style length. And between Yucca brevifolia and Yucca jageriana, we see actually quite striking and significant differences in style length, with Yucca brevifolia having a much longer style. And in fact, this distance from here to here is exactly the difference in body size between the two moth species. Or that is, this distance right here is equal to 1 Tegatecula synthetica, and this distance right here is equal to 1 Tegatecula antithetica. And indeed, if we look, let's skip that for a second. If we look across populations throughout the Mojave, what we see is that variation in population level differences in style length are strongly correlated with differences in population level variation in the moths in both body size and in ovipositor length. So again, it's pattern phenotype matching. And if we use a statistical analysis to compare morphological differentiation across Joshua trees, what we see is that indeed, although Joshua trees are significantly different from one another in a variety of vegetative features, such as height of first branching, and how many branches they have, statistically, differences in floral traits are much greater, and the greatest difference of all is in the length of the style, or that place where the female moth is going to sit during oviposition. Okay, so what we see overall in this system is that there's significant morphological differentiation between Joshua tree species. These differences in morphology are correlated with the geographic distribution of the two pollinator species. Floral traits seem to be correlated with moth traits, and the greatest morphological difference is in the feature of morphology that is most directly involved in the interaction. And we think, our hypothesis is that pattern that we're seeing is not a coincidence. Our hypothesis is that this is the result of reciprocal coevolution, reciprocal evolutionary change. So for example, we might, maybe people can see very lightly in there, there's supposed to be ovules right there. We might imagine that within Joshua trees, they may be selected to increase the length of the floral style and therefore reduce the number of ovules that are available for moths during oviposition. So by having a longer style, they're potentially going to lose fewer seeds from the Yakima flower. On the other hand, from the perspective of moths, we could imagine that on the one hand, there's selection for increasing body size and increasing ovipositor length to allow them to access more of the ovules. However, we also know from some experimental work that's been done by Deb Marr at the University of Indiana that moths who have especially long ovipositors and actually injure the ovules, they actually cut through the ovules during oviposition, that actually will prompt the flower to abort and die, killing both the ovules and the larvae inside. So from the moth's perspective, we think that there may actually be selection for matching to being exactly the right. And lastly, we suspect that this may actually be a case of co-speciation that's been mediated by pollinator selection. But we think we might have here the type of process that Fern Grant described, in which selection by pollinators might give rise to new species. And I've already kind of gone over the evidence for this, but Yucca vermifolia and Yucca chigeriana are each other's sister species. Tegaticula synthetica and Tegaticula anathetica are each other's sister species. Their morphological differences are greatest in features associated with the interaction. And because yucca moths are highly host-specific, this may provide a mechanism of reproductive isolation to go along with this selection, divergent selection. Okay. However, as I argued at the beginning of the talk, frequently patterns such as co-speciation could also be explained by extrinsic biogeographic history. And so to tell you a little bit about the phylogeography of Joshua trees, in this case, we suspect that speciation driven by riparians or speciation driven by the abiotic environment may actually represent a strong alternative hypothesis. So as Professor Riddle described to us yesterday during his talk about biogeography of warm deserts, we know that the Mojave Desert region was bisected by a large body of water, something known as the Booz embayment, which may either have been an estuarine extension of the Sea of Cortez, or may have been a freshwater lake system. And this existed between about 9 and 4 million years ago. And as Brett described with many more taxa than I was able to do, we know that there are many species that show a common break or a common phylogeographic pattern of this east-west split. So perhaps this is just another example of the same thing. So to try to look at this question, we need to start to ask what might be our expectations under a co-biparian scenario, or a scenario where biogeographic history was driving speciation. And in that case, I would expect that both the Joshua trees and their pollinators would show similar phylogeographic patterns, that the time of speciation should be about the same in both species if they're responding to a common biparian event. And if it's actually the Booz invasion that's driving this, the time to come ancestor, the divergence time, should be between about 4 and 9 million years. Okay, so to look at this question, we generated sequence data, about 2,400 base pairs of data from the chloroplast genome, and about 2,000 base pairs of mitochondrial DNA from the yucca moths, and about 500 bases of nuclear data from the gene elongation factor 1 alpha. We then directly estimated mutation rates using Mike Sanderson's program R8s. And then we took this genetic data and mutation rate estimates and fed them into Jody Hayes' program IM. And now just to look at some cartoon representations of the results, looking first at our moths, we see, as we expected, that Tegaticula synthetica and Tegaticula anathetica are strongly supported as each other's sister species, and each of these two species are reciprocally monophyletic, with the times of common ancestry of all haplotypes of about 5.5 million years. However, when we look at phylogeographic patterns within the host plants, within Joshua Tree, we actually see something very different. Firstly, neither of our two species is monophyletic. Both of these species are para or polyphyletic, and the time to common ancestry is quite a bit younger, at about 3.8 million years. However, these patterns on their own don't necessarily tell us about phylogeographic history for a couple reasons, and these have to do with the stochastic nature of the coalescent process. As both Dr. Avis and Brian Parsons described to us yesterday, it's actually quite common to observe paraphyletic species simply due to the retention of ancestral polymorphisms. And in addition, we know that the time to common ancestry of haplotypes should, barring dispersal, so barring gene flow, always predate the time to divergence. And in fact, sometimes it can predate it by quite a bit. So what we actually need to do is estimate in some coalescent framework the time to divergence and rates of gene flow between our two species. So to do this, we use Jody Haysberg and IM. Looking at the results first from the Yucca moths, what we see is in general a fairly well-behaved posterior distribution with the point of highest posterior probability indicating a divergence time of about 1.1 million years ago, or that is much, much later than the Booz statements. However, when we look at Joshua trees, the first thing we see is, wow, our posterior distribution is really crummy. Or that is, this posterior distribution is essentially flat over most of the ranges of values that we've evaluated. So we actually have very little power to estimate divergence times within Joshua tree. However, the one thing that we can say is that their divergence time appears to be significantly later, or significantly further back in time than the divergence time of the Yucca moths. Also, when we looked at our rates of migration between our two Yucca moth species, as you would expect for species that are reciprocally monophyletic, we inferred no gene flow at all. However, within the Joshua trees, we actually see evidence for significant ongoing and asymmetrical gene flow, with gene flow happening from Yucca vermifolia into Yucca jageriana. And it's unclear from these data whether this might represent sort of overall patterns of gene flow, or perhaps this is a case of chloroplast capture or plastid integration. So far, it looks as though Joshua trees and the pollinators do not show a similar phylogeographic history, and the time to divergence in the moths is significantly younger than the time to divergence in their hosts. And the moth divergence time seems to strongly rule out the Booz embayment as a potential vicariance mechanism. And for the moment, covariance seems unlikely, but it's actually difficult to evaluate this hypothesis given the low power of the chloroplast DNA to infer divergence times. Okay. And overall, this analysis has sort of left us really with more questions than we had when we started. So for one thing, there's this very strange apparent gene flow in the chloroplast DNA. We know that chloroplast DNA is uniparentally inherited, but it is passed only through the seeds. So perhaps nuclear data that would reflect pollinator-mediated gene flow might tell us a different story. If it's not the Booz embayment that's driving speciation, is it possible there might be some other common vicariance mechanism? And lastly, given that we see these high levels of gene flow between Yucca vermifolia and Yucca jageriana, why do they remain morphologically differentiated from one another? One possibility is maybe that the chloroplast actually doesn't reflect what's happening in the nuclear genomes. Maybe there are barriers to nuclear gene flow. Or maybe what we're seeing is that strong divergent natural selection is maintaining differences between species in the face of ongoing gene flow. Okay, so next, to try and continue and look at some phylogeographic patterns, as we've seen throughout these talks, we know that Pleistocene climate changes through Milankovitch cycling had major impacts on the distribution of many species, and in particular, it had significant impacts on the distribution of species within desert environments. And our time to divergence within our two Yucca moths is about 1 million years. Coincides approximately with the onset of major Milankovitch cycling. And in addition, so as I showed you earlier, Joshua trees have this sort of fragmented distribution currently, with these major barriers to dispersal being inhospitable habitats. However, perhaps at some earlier period, perhaps during a previous interglacial, maybe Joshua trees may have had a more contiguous distribution. And on the one hand, maybe fragmentation of that distribution through climate change might have driven speciation. Also, periodic range changes might provide opportunities for gene flow between our two Joshua tree species. And there's actually a good reason to suspect this within Joshua trees in particular. So we actually have extremely good paleodistribution records for many plants from within the Mojave Desert region. These records are coming from studies of the nesting habits of this animal, the pack rat, and the Yadama albivula. These are cache hoarders, so they will build these enormous nests where they gather together leaves and branches and sticks and bones and rocks and all kinds of things. And then over time, generation after generation after generation of pack rats will build a new nests on top of the old ones. And over time, this sort of sedimentation combined with the accumulated effects of lots of pack rat urine and feces creates these delicious sort of thick midden cookies. And what we can do is go and dig those out and separate out particular strata in there and then dissolve them in some really powerful solvents. And then what we get out is a record of what types of plants were growing in a particular area at different points in time. And we can actually carbon-14 date these to know exactly how old they are. I can say we here, this is we in the like Royal Science we. This is all work that's done by Julio Betancourt, Tom Van Deventer, and others in the 1970s and 80s and early 90s. Okay, but from these data then, we can actually reconstruct a record of the paleo distribution of Joshua trees. So in this picture here, all these cross hatches, that shows the current distribution. And all these little triangles down here, those show paleo records from the last glacial maximum. And what you can see is that 21,000 years ago, there were large areas that were formerly occupied by Joshua trees, including sites down on the Sonora, Arizona border, and sites as far east as Tucson, Arizona. So we have good reason to think that there have been changes in distribution associated with glycemic climate change. And so to try to look at this question, I collaborated with my colleague Will Godso, you'll see a picture in a second, to do some paleo niche reconstructions to try to predict past distributions. And our predictions for this are simply that maybe at the last glacial maximum, Joshua trees may have had a more contiguous distribution that might have allowed for either gene flow or perhaps climate change may have been a mechanism of speciation in the past. And also, given that we have this paleo record, if our niche modeling is doing a good job of predicting past distributions, we should be able to see a match between our predicted past distribution and where we know Joshua trees occurred in the past from the pack rat record. And so to try and build this model, again, I've collaborated with Will Godso, who's formerly a graduate student at the University of Idaho. He's now a fixed-term faculty member at the University of Canterbury, New Zealand. And what Will did was to take a fairly extensive database containing about 5,000 GPS records, known occurrences for Joshua tree. He then sub-sampled those and combining those with data from BioClim, he builds a niche model, which he then projects onto current climate distributions to predict its Joshua tree's potential current distribution. And then using a subset of the presence data that are not included in building this niche model, we can actually test how good a job our predicted current distribution does of saying where Joshua trees could occur. Similarly, to build a paleo distribution, we can do the same thing. We can take a subset of the data, combine it with BioClim data, build a niche model, and then use paleoclimate reconstructions and project that distribution back into the past. And I think this is really cool. We can take those midden records and then ground truth, which apparently is the word I'm not supposed to use, apparently, Frank, but we can truth those.
I like them.
Okay, we can truth those against the midden records. Okay, and so, for example, here's one of our sets of niche reconstructions. Up here is the distribution under the current climate regime. All these hatched areas are where Joshua trees occur currently, and these sort of lighter gray cells in the background are where we could predict potential distribution, or where the niche model says Joshua trees could occur. And what we see using an area under the curve statistic is that our current model actually does a very good job of predicting current distribution. And down here, we've taken that same model and projected it back to the last glacial maximum, and then compared that to our midden records. We see that here our niche model does an acceptable job with an AUC score of 0.8. For reference, a score of 0.5 is no better than chance. It's like you're guessing. And we also see that there's a much more contiguous distribution at the last special maximum. And so I was very excited about this result, but unfortunately, Will Godso just can't leave good results alone, and he decided that he should go and look at how sensitive these results were to alternative mechanisms, the alternative processes for niche reconstruction, and alternative mechanisms of threshold for deciding when, at what point the probability of presence is high enough that we say yes, there's a Joshua trees there. And so here, this is a current niche model built using maximum entropy. This one here is built using boosted regression trees. This is built using maxent. And this model also does a fairly good job of predicting current distributions. In fact, it's slightly better than the boosted regression tree models. And it does an acceptable or a fair job of predicting our paleo distributions. So even though however, even though these two models seem to be performing about equally well in reconstructing paleo distributions, our results are about as different from each other as they could possibly be in terms of what I'm interested in. That is, this result actually predicts a distribution that remains fragmented at the last special maximum, where there's no opportunity for gene flow across this same central Mojave divide. Also, just a sort of trivia note, this model actually also reconstructed very high probability for presence of Joshua trees in northern, in Baja California Norte, in the Sierra Juarez. And I hear from Brett Riddle that there's a forthcoming paper from Julio Betancourt's lab looking at PACRET hidden records here, and I'm very excited to see if they find Joshua trees there or not. We'll see. Okay, but so looking at the results of this phylogeographic analysis, what we find is that although our paleo niche distributions do a pretty good job of predicting known past distributions, model uncertainty actually makes it very difficult to distinguish this possible scenario from this possible scenario. So overall, unfortunately, phylogeography seems to be letting me down. In that, perhaps because of my crummy Cloverfest data set, there's not very much that we can infer from the molecular data, and similarly, model uncertainty in the niche modeling makes it very difficult to infer what might be potential past distributions. And so, in some ways, out of frustration, what I've tried to do is instead of trying to distinguish these two particular processes by looking at evolutionary patterns that might be predicted by both, I've instead tried to identify what are some evolutionary patterns that might be unique predictions of coevolution. And one of them that I'm talking about right now is that coevolution actually predicts local adaptation. Sorry, I'm just looking at my time. Okay. And to do that, we've been using some tests for reciprocal natural selection. And we've been doing this, so earlier, I showed you this map distribution. Some of you may have noticed this star up here at the top. That's a site in central Nevada, actually just immediately outside of the Nevada test site. They set off on the bombs. And at this site, we have a zone of sympatry where both of our two Joshua tree species and both of our pollinators co-occur together, often within meters of each other. And this zone of sympatry actually presents to us an exciting natural laboratory where it's possible to do some manipulative experiments and some population genetic tests that would not be possible elsewhere. So at this site, one of the things we've been doing is simply a reciprocal transplant experiment. We're transplanting the moths of the trees, in which we collect either Tictica synthetica and then introduce it onto the caged inflorescences on either its native host or on its non-native host. Similarly, we can take Tictica antithetica and do the reverse. And so to complete this experiment, our predictions here are simply that we should expect to see if there actually is coevolution and there's local adaptation, there should be a strong host by pollinator interaction on fitness. Or that is, how well each moth and each tree does is going to depend upon which species they're interacting with. And so we should expect that trees should have lower fitness when they interact with the wrong moth, and plants and moths should have lower fitness when they interact with the wrong tree. And so to complete this experiment, we simply go out in the spring and identify trees in the bud stage. We like to call it the artichoke stage. And then cover these with some cloth mesh. We then place some chicken wire over this to keep out rodents, and actually cattle are a major source of predation on flowers. We then collect moths, sort them in species and in scope, and then experimentally introduce them into our exclusions. We can come back later in the field season and collect fruit. We can then rear out larvae, and using microsatellite genotyping, we can actually assign these larvae into natural lines, so we know exactly how many larvae are the product of each female that we introduced. And then we can also dissect out the seeds and count the number of seeds that were produced and the number that were eaten by Yakima. And so what we see, first looking here at some larval data, is that overall there's actually a very strong host by pollinator interaction, or that is both species of Yakima have much higher fitness or much greater success when associated with their native host. And this is work that was done by an undergraduate student in my lab, Tom Holloway. Secondly, looking at fruit sets, or simply how many fruit are produced, we again see a strong host by pollinator interaction, in which each species of Joshua tree is producing more fruit when it's associated with the native pollinator. And lastly, when we look at the number of surviving seeds, so the number of seeds that are produced and don't get eaten by Yakima larvae, there is a suggestive but not significant host by pollinator interaction, suggesting that perhaps the trees also may have higher fitness associated with native players. So overall, we see a significant host by pollinator interaction. Pollinators seem to show a significant local adaptation, and the trees show a significant, suggested an unsignificant trend. So these data seem to suggest to us that there may be coevolution, and there may be selection for phenotype matching, but it's not clear from these data what is actually producing that local adaptation. Why is it that moths do better on the native host, and why is it that plants do better? However, we're continuing to try to explore this with some additional experiments. And one of the things that's exciting about this particular study site is that at this area, we actually see continuous variation in styling, or that phenotype I showed you earlier, which we think is important in mediating the interaction. We can find trees with very short styles, trees with extremely long styles, and trees exactly in the middle. And so it's possible for us now to look at these phenotypes as continuous variables. And just some preliminary data here, as we look at seed survival, or the number of seeds that are escaping eaten by Yakima larvae, that seems to be very strongly associated with styling, with trees with intermediate styles actually having the lowest fitness, or the greatest rate of loss of seeds to Yakima larvae. And we're continuing to try to expand on this experiment using variation in the moths as well as variation in styling to create all possible combinations of moths with very long ovipositors on trees that are medium or short or long, etc. Stay tuned. And lastly, sorry, I'm getting a bit slower than today. Usually I get so nervous that I race through everything. So probably just going to be as well. You're at the end anyway. Okay, I want to tell you a little bit about some population genomic approaches or genetic approaches that I'm trying to use to quantify selection in this system. And so again, what we've been trying to do here is identify patterns that may be uniquely predicted by coevolution and not by biogeography. And the pattern that I'm looking for is something called islands of genomic divergence. Or that is, I'm looking for places in the genome that are much more differentiated than the rest of the genome, and asking, are those places that are involved in mediating the interaction? And so, as I mentioned to you earlier, Tickford Valley presents this really nice natural laboratory to us. For one thing, we have both locally allopatric and locally sympatric areas occurring over a very small spatial scale, so we can more or less rule out climate as a potential confounding factor. And in addition, at this site, we actually have very clear evidence for hybridization, or that is, within Tickford Valley, we can find individuals of every possible genomic background, from trees that are 100% Jageriana to trees that are 100% Bronifolia, and every possible combination in between. And this allows us to do something called admixture mapping. If you want to know more about admixture mapping, we'll talk afterwards. And basically what I'm doing here is combining two different approaches. One is called genome-wide association studies, which is simply identifying statistical associations between genotypes at a particular locus and the phenotype we're interested in. and a second approach called FFT outliers to identify places in the genome that are associated or seem to be experiencing natural selection. Okay, and so again, looking at predictions, if coevolution is what's happening here, we should expect that most of the differentiation between species should be associated with genomic islands or small regions of the genome that are associated with styling. And secondly, we should expect that styling shows evidence of being on inversion natural selection. Okay, and so just to kind of briefly overview the methodology of a genome-wide association study, all we're doing is looking at variation in phenotype amongst individuals that have a particular genotype at a locus. So for example, these might be individuals that are homozygous for, say, a Hungariana allele or AA allele, and looking at the distribution of styling, looking at heterozygotes, and looking at homozygotes. And in this very simplified example, you can see that there's sort of activity. Each additional brevifolia allele causes an increase in styling. And then what we do is just to complete a series of linear regressions in some statistical software. Okay. However, in order to do this analysis, you also need genetic data, it turns out. And so to do that, we've been using a next-gen sequencing-based approach called RAD, or restriction-associated DNA. RAD is essentially like AFLPs, but you would have actual sequence data from it that allows you to assess homology of co-migrating bands. You could also think of this as a complexity reduction step. We don't want to look at the whole genome because that would be impossible, so we're just looking at things. Okay, and so what we do is we generate these small 100 base pair sequence reads from regions that are associated with restriction sites. We then pass those through a program called STACK, so we developed at the University of Oregon, that's going to group our reads into putative loci. We now genotyped about 147 individuals, which generated about 2.2 million RAD tags. The vast majority of those are unique, or they're present in only one individual. So we then narrow down to only those tags that are genotyped in at least 90% of our trees and that are actually polymorphic with contained variation. We're left with about 5,000 loci containing about 16,000 SNPs. And then we just measure association between the genotype at each locus with phenotype in R. And so here I've actually restricted my analysis to a mere 700 loci, and focusing on those loci that are biallelic. They have only 2 alleles, and they have no more than 2 SNPs per locus. And the not very good reason for doing this is that those are just easier to recode, so I can put it into R. And from that result, we see that there are actually 5 loci that show significant association between genotype and phenotype after applying a very strict Bonferroni correction. And so, for example, this locus, 6125, shows a very strong pattern of additivity, where each additional brevifolia allele adds about 1 millimeter of styling. And this locus alone explains about 30% of the variation in styling. Okay. So now, having identified regions of the genome that appear to be associated with variation in phenotype, our last question then is, are these regions under selection? And to do this, we use an approach called FST outliers. It's becoming extremely popular in population genomics. And so I think most of the phylogeographers already understand FST, but just to briefly remember, FST is just a measure of genetic differentiation between populations at a locus. So an FST score of 1, for example, might represent 2 populations that are fixed for different alleles at a particular locus. Everybody in population 1 has A, and everybody in the other population has T. An FST score of 0, on the other hand, might represent 2 populations that are identical in allele frequency. And typically, when we look across the genome from locus to locus to locus, what we see is that most loci cluster around some central genome-wide average FST score, which represents the balance between the divergence through genetic drift and coherence through gene flow. However, occasionally, we can identify a few loci that are either much more differentiated than average, potentially representing the effects of positive selection, convergent natural selection, and some loci that are much less differentiated than average, potentially representing the effects of balancing or negative selection. Okay, and so again, we're taking RAD-seq data that's been cleaned by stacks. We pass that to populations, which is just a built-in population genetic software in stacks, or to Harlequin. And so first, just looking at some overall summaries of the data, these are all those 16,000 SNPs I mentioned earlier. What we see is actually quite surprising, which is that over almost the entire genome that we've surveyed, the FST score is essentially 0, or there is no meaningful difference between yucca vermifolia and yucca chieriana. These loci are not places that where there's no variation. Remember, this is only polymorphic loci, but typically these are places where there's maybe one rare allele present in both populations. And then we see this very, very long tail. And out here on the right, there are a tiny minority of loci that are very strongly differentiated between yucca vermifolia and yucca chieriana. Okay. So next, I analyzed these using an application that's built into Arlington. And essentially what this is doing is just controlling for the fact that the potential value of FST depends upon the overall allele frequency. And so what I'm doing is I'm plotting for each one of these dots as one locus, and I'm showing its FST score of that locus versus the expected level of heterozygosity if we cross two populations together. If that doesn't make sense, just close your eyes and go with me. Every little spot in here, so the black ones, those all represent loci that do not appear to be significantly different from the genome-wide average. Each one of these blue spots up here above this dotted line represent loci that have especially high FST values, or perhaps show the effect of positive selection or divergent natural selection. And all of these loci down here show loci that have very low FST values, potentially representing negative selection with the same allele being favored for populations. So what we've done now is just to map onto this. Sorry, skip that. Those loci that were significant GWAS, remember we found only 5 of them. Of those 5, 3 are significant FST outliers, and all of them show very high levels of FST, potentially suggesting a relative selection. However, I wanted to, rather than just point at this figure and be like, look, hurry, selection, I wanted to actually quantify it statistically. So what I did was to apply a much more generous significance cutoff. So I've used a significance value of 0.5 here, and I've done that simply just to give me enough counts in the 2x2 comparison that I can analyze it using the square test. And what we find is that overall, those loci that are associated with styling are much more likely to be FST outliers. Or that is, those regions that seem to be affecting variation in form morphology are much more likely to also be under natural selection. I'm continuing, by the way, to look at this using some genomic clients analysis, which fortunately for Frank I cut out, otherwise it would have been an hour and a half. But what these data seem to tell us is that genome-wide yucca vermifolia and yucca chieriana are extremely similar to one another. Styling genes are more likely to be under selection than the genome overall, and in allopatry, so the features of allopatric sites are styling appear to be under divergent natural selection. Future plans are continuing to follow us up and now ask, having identified these putative styling genes, might those be speciation genes, or they concord with species boundaries? And we also want to use some coalescence-based approaches to ask, when did natural selection act on these genes? So if these genes start to diverge at the same time that our two yucca species diverge. So in closing, our phylogeographic work seems to suggest that speciation is inconsistent with known vicariance events. Our speciation system could be explained by placing climate change. However, directly testing for selection, that is identifying patterns that are uniquely predicted by coevolution, has given us some clear results. That is, we see patterns of local adaptation that seem to be associated with variation in styling or variation in our phenotypes, and our population genomic analyses suggest pollinator-mediated selection acting on styling. And in closing, 5 minutes left, right? Okay. I just want to thank the many people who contributed to this work, especially my postdoctoral advisor, Will Premier, and his graduate students at the University of Idaho, as well as my collaborator, Matt Streisfeld, my incredibly talented technician, Laura Flatts, my many undergraduate students, and of course, the ubiquitous funding agencies. Thank you very much.
Gracias, doctor Edwin, por su plática. Tenemos 5 minutos para preguntas.
I have a question about the first part of your talk, maybe.
About the geography or about the
Yeah, the coevolutionary study. It's that I think you found 3 phyllo groups with the chloroplast data.
Yes.
And I think Joshua tree species are dispersed by scatter hoarding rodents.
That's the hypothesis, yes.
And maybe that chloroplast signal you found is more related to the interaction between the dispersers than the interaction between the pollinators.
Absolutely, I absolutely agree. So one of the possible explanations for the discord that we see between the pollinators and the chloroplast data is that the chloroplast is being parentally inherited in seeds and not in the pollen. We would really like to be able to compare phylogeographic patterns based on nuclear data, which might reflect to us what's happening in the nuclear genome with the pollinators. My expectation is that if we could do that, we would actually see divergence times that are much more similar. However, as I was talking to somebody last night about, it turns out nuclear gene phylogeography in plants is hard. I am hoping, though, that the RAD-seq data may allow us to answer that question. We're just not there yet in terms of data collection, but I think that's the next thing. Does that make sense?
Yeah. Yeah.
Thank you.
Here, I wonder of something that you didn't mention, of the convolution description of your moth and yucca flower. Okay, what can you tell us about natural enemies of the insect? Maybe that could influence the multiple prospect that you have. I don't know. I'm just wondering. But if you have to detect something, maybe some parasitoid or not, that are part of the third trophic level normally that we know exists in every interaction.
Absolutely. So indeed, there are parasitoids in the genus Stegogastra that O-positive non-methylmethylene. I'm not as familiar with that work. My colleague, David Althoff at Syracuse University, has worked on that some. And what he seemed to find is that actually the parasitoids seem to be quite generalists, so they are associated with yucca moths, but they O-positive on a variety of different species. It is, of course, possible that there is cryptic species diversity there. So just like work that's been done in Costa Rica, looking at parasitoids associated with various lepidopterans. In that case, they found all these cryptic mitochondrial lineages, and I just don't know whether that's been done yet. I will say that in my experience, the parasitoids are actually quite weird. It's pretty uncommon that I see them, and they're very noticeable. They're fairly large and bright red. I just don't see them. It's kind of a dodge to your question. Yep, could be.
Chris, I was wondering about the temporal incongruence between the ocean trees and the yucca moths. And I was wondering whether the ocean trees at the population level can go through time through, I don't know, self-pollination or some other, other strictly dependent on cross-pollination.
So, I'm going to give you this unsatisfying answer. They are potentially self-fertile if we cross the trees to themselves. And in fact, one very surprising result from the reciprocal transplant experiment, which I didn't talk about, is that we actually went through, we took the seeds resulting from the reciprocal transplant experiment and genotyped them, and we actually find that fairly often, when we put moths into a cage, they're actually pollinating the tree with its own pollen. However, in nature, according to work that's been done in Yucca frondosa, when there is self-pollination, those fruits are poor competitors with other developing fruits, and as a result, flowers that have been outcrossed are more likely to set fruit. I think it may just be in this very controlled environment where we prevented all the natural pollination that we see that self-pollination or self. It is possible, I suppose, that that may be an alternative explanation for the differences in the phylogeographic patterns, and I would have to think a little bit about that. But I suspect that, in fact, primarily in nature it's not this. Does that answer your question?
We can talk
We can
about
talk.
it later.
Thank you very much.
Yeah. Restart or the mirror? Just restart. Really? Hold on. We have to. It's right here, the mirror. You restart. No, it's not. So then just be sure that'll work. Yeah. Yes, do. And also, I think it shows how I know that sense. That's what our field needs to do. To break it down. Yeah, so you take that out. Should go back. It's really impressive. say things that we can Yours is a good one. Good thing.
Oh, hey! I've never felt anything. All right.
Well, that's a price break. So that's not what I'm saying.
Are you ready?
Tenemos el honor de presentar al doctor Frank Burbury, quien es profesor en el Colegio de Estudiantil, en donde a través de herramientas moleculares y estadística fenogenética busca describir y entender los procesos de especiación e historia evolutiva de especies de reptiles y anfibios, así como los procesos y eventos geográficos que promovieron la diversificación de dichos grupos. Démosle la bienvenida al doctor Frank.
Thank you. Hey, was she telling the truth? I could. She was, okay.
Man, I hate being in front of a mic.
All right. Well, thank you very much for inviting me. Thank you, Inicol and the postgraduados and all the niños and niñas here. And, thank you, the professors, the opening bands, everything. Very, very happy to be here. For me, I'm a snake guy. I'm an evolutionary biologist, but really I still always be a snake guy, so I'm kind of very close to paradise for snakes, right? And I'll tell you a real quick secret. All American U.S. herpetologists are very secretly, maybe openly, very jealous of Mexico. Very jealous. And I'm thinking maybe we could start trading states. Like, you guys could have North Dakota, and we could have Veracruz. No! North Dakota's wonderful. Bison, big grass one. Okay, all right. So, all right. Well, my talk is the best one here, and basically, I'm going to try to tie up a lot of the concepts that we've talked about, but more in terms of a focus on something that I'm very interested in and have been interested in since I was a kid, and that's communities. And not interested in evolutionary processes, things like that when you're a child, but when you live in, I grew up in central Illinois, which, if you're a reptile fanatic, is about the worst place in the world to grow up, right? Maybe the moon would be slightly worse. But I had relatives that lived in southern Florida, so I would look around in central Illinois and I'd find 2 or 3 species of snakes, and my brain would just be on fire. Why aren't there 40 species like at my aunt's house? You know, and why are these 3 species also found in my aunt's house? And I would think about that, but I had no idea how to solve it. So I'm taking these questions, from my childhood and trying to figure these out in adulthood, right? So I guess maybe I'm still a child. I guess I dress like one. Somebody told me I dress like a 12-year-old. So just in a nice thing. But anyhow, so we're going to talk about community structure, but in a very sort of broad context, from broad to very narrow. So communities, right? Communities could be any spatial area that you want to define as a community where species would interact. And it could be communities of, and what I'm interested in, are like things that are similar in those kinds of communities that would be interacting, okay? And now obviously this has a temporal element to it, how communities are formed, and a spatial element. I'll go through all of this very nicely, I hope very nicely. And then interaction, species interaction. And all of these pieces will help form communities, okay? Now, this is a really famous figure, as famous as things are, I guess, in evolutionary biology. Let me see where this, the red button. Oh, yeah, I guess I should have pointed out myself.
able to get here, if you can believe that. And this scale is very a visualization. This is what I was trying to answer in Brett, which is there's a lot of feedbacks in these scales that you can't see. So if you look at this as a spatial scale here on the x-axis, and you have large space here and very narrow or small space here, small time here and large time here. We think of big processes at this edge of these graph, which is biogeographic processes, things like speciation and extinction rates, right? And then ultimately you're producing species and they're dispersing around the world, dispersing through continental systems, and something is allowing them to disperse or not allowing them to disperse, such as, and we call this thing environmental filtering. If you don't have the right kinds of characters to disperse through an area, or to arrive or stay at an area, or even to remain in an area, you're going to be gone from those places. And as space and time gets narrow, and you have these characters that allow you to exist in these narrow communities, those characters that allow you to exist in those communities can bring you into competition with other things that have those same kinds of characters that allow them to exist in those areas. And this was one of Darwin's paradoxes, okay? And so you can get these density-dependent interactions that exclude species from co-occurring, okay? Now, as I mentioned this Dan Warren paper in Triad, I'd like everybody to read it. It's kind of what we've all been thinking about, but there's definitely these scales, maybe slightly artificial and slightly conveniences for us to deal with. I'm not going to get so much into that, but I want to talk about some of these things. And clearly we have a temporal component to biology. Not everything that has existed will always be there. This is actually from one of my favorite weird websites, which is they take classic paintings of dinosaurs and put religious figures with them. For whatever reason, they've never explained why they put these on. So there's clearly a temporal component. Not all the species that have ever lived will be in a community. I know that's an absurd example, but I think it's just on the sort of way of thinking. And then there's a biogeographic component. This is a painting of the Garden of Eden, right? So there's not everything that exists on the earth right now can exist in one community. And I'll give you an example of that. Look, they're all getting along. The snake hasn't formed yet, and I can tell you the serpent is not the bad guy in the Bible. It's a loser, because they always talk about him having legs, okay? So if we want to look at this, then my understanding and what I want to tell you guys about is this community composition, that you have things like diversification, rates of speciation, rates of extinction. That's providing your source pools, your continental or global source pools for things that could be in communities. You have your biogeographic processes, things that allow, processes that allow organisms to disperse and move around the globe and to be in communities or not be in the communities. Then you have the ecology pieces, things that having to have traits that match ecologies to be able to disperse or land or survive in communities. And then finally, species interactions at the community level that either exclude or potentially promote species to being there. And so this talk is going to be linking short-term processes with long-term processes, okay? And so, by the way, all of these photos are mine of all the snakes that I catch all over the world. So, so the species interactions, we think of things like ecological filtering. That is selection for traits, right? If you're going to be where I live in New York, if you're a snake, you have to be able to hibernate. So you have to have those traits that allow you to go dormant for 5 to 6 months out of the year. That's a tricky thing physiologically. So that's an example of ecological filtering. There may be a ton of tropical species that never develop that and never will land in those kinds of areas. There's negative interactions. If you have all of these things have the same kind of traits to live in these areas, it's extremely possible that it puts them into competition unless they start diversifying on other axes so that they're out of competition. And so that again is the Darwin's paradox, right, for communities. And then you can just have purely neutral processes. That is, if you speciate and you're there or you're in an area, diversification, maybe you just get there by random dispersal, right? And you have to be in it to win it, right? So you can't go extinct. So it could be completely neutral, and that these processes aren't really doing much for community assemblage, or that the community assemblage is so unstable through time that these things are actually hard to see on a temporal scale, okay? So why the heck should we be working on snakes? Well, that should be obvious. They're the best, okay? They're super diverse. Most snakes, 96% in the New World, are super young, right? On the time scales. Everybody loves anoles, right? Everybody looks at anoles for diversification. Those are old fogies. Those things are old. These guys are only, the colubroids are only around 30-some million years old, produced over 3,000 species, conquered every niche, occur in every continent except, obviously, the very coldest one, right? And they do this without limbs, okay? And they're extremely diverse, morphologically, right? Head shapes, productions of venom, living in super arboreal ones that never come to the ground, super fossorial ones that never go above the ground. Diet, eating everything from huge mammals to other snakes to small little bugs, lots of diversity, right? And they occur in large communities. Often, even though in the tropics you don't see many snakes, they're going to be the largest component of your squamate community. Squamates are lizards and snakes. You'll see all the lizards because they're happy things that stand out and sit on limbs, and you can go and view them and love them. But these guys are hiding, and they're everywhere, and there are more of them. So they're super diverse. And they have massively strong ecological gradients, okay? As you go away from the equator towards the poles, you drop in species. And that's a clear pattern with most organisms, but it's very strong, and it's very strong even in the United States. And so if you take a survey of people's favorite vertebrates in the United States, it's snakes. At least if you take that survey within my lab, okay? Anyhow, so I live, unfortunately or fortunately, in New York City, but I have this wonderful house up in Woodstock, New York. Woodstock, New York, where the hippie, concert was. Actually, not really at Woodstock, somewhere else. If you want to go up there, it's like going to a Disney World of hippies, and you see a lot of elderly hippies hanging around, and it's pretty bizarre. But in the forest, there's large forests there. You can have, this is moderate, 11 or 12 species of snakes, but they're extremely different in this area, right? And so you have a community that displays a lot of differences. For instance, on this side of the bar, you have all egg layers. Here you have all live bearers. 2 venomous species that feed on mammals. These guys feed exclusively on fishes. These feed on newts, toxic newts. Oh, you really want to do this? You can't hear me? Oh, okay. Oh, okay, all right. I feel like a motivational speaker. Okay. You have things that specialize on slugs, insect eaters, all kinds of stuff in a small northern community, right? And the sizes are really different. You have this beautiful storaria. That's not a snake, that's one of my grad students holding a giant rat snake. So massive differences in size, diets, right? Eating all the things other people like. People really hate this, of course, squirrel going down. That's the way it is, birds, insects, and lots of specializations even in just these small communities. And I'm just pointing out why I think they're good models, right? Not just to bore you with snakes. You have lots of live bearers and moms hanging out with their little babies, and egg layers as well. And you have diurnal species and nearly strictly nocturnal species, and ones that shift back and forth, okay? So just really quickly, if you do snakes in North America, at least the US here, the origins are about 110 million years old, okay? And in the US, there's 164 species, which represents only 23% of the families and subfamilies of the world, 5.4% of the global diversity. And if you go to a single best largest community, you get about 1.2%. And if you include Mexico on this, the numbers are reduced just as well. But what that shows is that obviously globally, the source pool of what could be in any communities or even regions are limited to what's on the planet Earth, right? That's your first, dumb guy observation, right? And I think it's an important dumb guy observation, which is not everything occurs everywhere, okay? And it gets smaller and smaller. So the point of this talk, at least the workflow, is I want to try to understand that community development in some way. And first I'm going to look at the origins of these groups. How did they get here? Why are they even here in the New World? First, I'm going to use these data phylogenies using next-gen data. Okay? We're going to then assess this global biogeographic patterns of how they got here. Once we know who our source pool is, we can ask very cool questions, I think, about biogeography. We can ask the question, if they all got here at the same time in the same area, should they show the same sources of ecological opportunity? Should they show extreme diversification and niche filling, right? So that would be one of the predictions of having a temporally and geographically same area of origin. And then we're going to look at assemblage, right? And how do these feed back into regional biogeographic community assemblage, things like beta diversity, et cetera. And then we're going to take together species, traits, environment, ecology, and phylogeny to look at the mechanisms that may or may not allow these communities to form over time and space. So I'll break it down. I think I'm going to break it down pretty easily. So global biogeography, right? What we need are species, genes, trees, areas. And the question that I'm going to ask is, for the New World, do these organisms have the same area of origin, the same timing of origin? And if you have these, will you predict that they have the same process of diversification into these areas in the New World? Okay. Now we're going to look at 6 major groups, and these are the most diverse groups in the New World. These represent about 96% of the species. These are colubroids, just to familiarize yourself with them. This is a nature scene. These are your water snakes and garter snakes. They bite and defecate on you. They're not very nice, but I love them. So, it's like bad children. You still like them. So 60 species. Colubrians, these fill a lot of niches, from your beautiful Drymobius here, et cetera. And about 120 species. My favorite group that I have are peltonines. So these are the rat snakes and the pseudolapid that I'm dying to see, that really only occurs in this region. And the dipsodines, which are massive. In the New World, it's ringneck, or in North America, it's generally ringnecks and things like that. But when you get to Central and South America, they fill every niche. So it's around 730 species. Our beautiful elapids, the coral snakes, around 70 species, and then our mighty viperines, and that should be about 130 species in the New World. So these are our main groups. Now we're going to generate trees, and I built a method. I teach this at Nesset. We're going to be teaching it at the evolution meetings a week before the evolution meetings, hopefully in Brazil. And it's open to anybody who wants to apply to come. So it's next-gen methods and computational methods for answering questions in phylogenetics and phylogeography. So I built one of these methods that can take, and in this case I have 304 loci for all of, I'll give you an example of the rat snakes, all 90 species of the rat snakes. So take all of these loci and you generate gene trees from these, and you can generate 100 bootstrap trees. You have error with that, or you can generate posterior probability distributions, all automated. And then you take all of those, and from each slice of each gene tree across each locus, you can ask, what are the species trees, right? And here I'm using a quick method, the MPS, maximum pseudo likelihood estimator by Lane and Liu, to build species trees really quickly. There's a couple of other ones, small RF. I always tease Leanne and Liu. You didn't grow up in the parsimony era where you had real bullies and parsimony guys, because if you did, you'd never name anything MP, maximum parsimony, that means maximum pseudo likelihood. Anyhow, so you build all of these species trees, and then from these you can make a consensus, right, and generate your single species trees with lots of support. With these, over all of these snake groups, I get phenomenal trees, phenomenal support for most of the nodes. And if you just really want to look at this quickly, well, what do we get? If we look at all 90-some species of rat snakes, and this is with my doctoral students in Chen, what we see are all the different species tree methods. This is RF distance, so this is a measure of topological difference, right? So this, and we converted it into percentage. So all the species tree methods are giving you very similar topologies for the species trees. If you compare the species trees to the concatenated, you get close to 50%. So it's 50% different at that point of the total RF. And then if we look at our own tried and true mitochondrial trees, which I've been hanging my hat on for half of my career, we see it's much higher. And then if you look among the distributions just of the loci by themselves, these are the among gene tree loci, just comparative among themselves. They don't agree at all. So most of these big anchored methods produce lots of gene trees that do not look like each other, right? Some will support one small clade, some will support some other one, and some will have just mainly no resolution for most of the tree. And that's why it's a pretty good idea to put this into a species tree context. So now if we use something like biogeovars to estimate ancestral area on these guys, on these big trees, we can see if we code biogeovars as a method for estimating ancestral area, okay, using likelihood and using estimates of dispersal and extinction, two confounding variables for estimating ancestral area. And we code these cool colors here for old world taxa, and we code the red and orange for new world taxa. We see pretty much what I got in 2007 with one mitochondrial gene and two nuclear genes, which is you have this jump from the old world to the new world and diversification in the new world. And you have that at the top of the mice. Okay? So that's a pretty clear pattern. By the way, you guys know BioGeoVaries? It's the strangest written R program on the planet Earth. And all of the help, if you type in help for R, it always just says go Bears. And I have no idea why he has. And it's not even the Chicago Bears, right? It's a California team, man. Yeah, the Cowbirds. So anyhow, the guy's a really nice guy. But this is a common pattern now. So I'm looking at, if you look at this is an example, these are the Viterids, this is the water snakes, the rat snakes, and these are skinks, right? Skinks because we had them. So you see this as a common pattern. They all have this Asian and North America distribution, and it's all around the top of the Miocene if you look at the distributional age for that day. And when you reconstruct that, the only area that was open is generally through Beringia. Okay? Now this is a really cool pattern because it suggests, at least in North America, that the species originated in the West and then dispersed to the East. And when I relook at this, just biogeographic analyses, I'm not going to repeat these, just inside the New World, you see this pattern. So this is a pattern to remember. Origin in the West to the East, West to the East. Oh, it's for all of these groups, okay? So this is 96% of the snake species in the US. So at the top of the Miocene, through Beringia, west to east, and 6 lineages out of 20 total main snake lineages that could have occurred from the old world. 6 maidens. The other ones may have made it, they're gone. Okay? So those 6 main lineages. So that sets us up for our stage. That's our species pool, and that's how they got here. Okay? So now we should want to know if they all got here roughly the same time in the same area. Do they show the same processes of diversification? Now, diversification gets batted around from phylogeography to old stuff to whatever. And I'm going to try to define diversification here in a very sort of species extinction oriented way. And this is from a fantastic paper, Sloss, 1950. This is the first guy to take fossil record and use differential equations to predict speciation and extinction rates. And the guy is amazing, man. So, I mean, I just try to think of this like doing this stuff in the 1950s with a paper and pen. So, with a paper and pen. So, diversity here is speciation minus extinction. And if you're at level 1, your diversification is increasing. This line is speciation, so your speciation is increasing rapidly. That's the part we always think of as an adaptive radiation. Species get into an area and they burst in speciation and fill niches. So it's not goal-oriented, it's just what happens. Okay? When you hit apogee here, this is when speciation equals extinction. That is, the rates of speciation and the rates of extinction are the same. This is the turnover frequency, right? And this is the fate of pretty much all groups, right? At least in the fossil record, which is extinction eclipses speciation and things go extinct. And so it's over, okay? At some point, humans will be at this level and the world will rejoice, right? So, question, is this an adaptive radiation? Is adaptive radiation deterministic given region and time? That is, if we know these groups got here in the same area, same region, same time, can we predict if they have the same diversification? And it's really quite easily, and there's been a lot of math in the last 4 or 5 years developed around doing this. Some more advanced here. One of the easiest ways is just take your phylogeny, and each node on phylogeny is a branching time estimate. So that's your time for estimating speciation, right, or diversification. Long branches can potentially be caused by extinction or delayed speciation time. And this equation has been known since the mid-40s, but has been updated for phylogeny. So if you want to predict birth or death rates through time, all you have to do is solve this equation given these branching times, okay? And what you're probably used to seeing is things like log species curves, right? And on a semi-log plot, that's log, and that's time, what you see is an even straight line. And that indicates that speciation and extinction have been the same through time, okay? And that's your typical birth-death model, right? If you have no extinction, that's a Yule model, right? So these are pretty easy to do. You can have your variable rates. Now, this is what most people think of as a signature of adaptive radiation, which is you burst early, so speciation accumulates or diversification accumulates early, speciation is really high early, and as you start approaching this carrying capacity, speciation declines, and it's easily defined by the number of packs at any one point given a carrying capacity, okay? And this can be dealt with in extinction. The old ways they didn't deal with extinction well, but they do now. So basically what happens is diversification or speciation declines over time as you approach this carrying capacity. So that's kind of what we expect for an adaptive radiation as niches are filled. And then you have a whole bunch of other null models. And some of these old null models is that speciation will decline with time as linear decay, but not because of a carrying capacity. That is another variable that you cannot predict. And speciation rates can shift dramatically too, such as when you have major changes in glacial cycles, right? If glaciers come down, you limit area, and speciation isn't going to be as high in these areas, okay? And so with that, we have a whole bunch of other models. I'm not going to go through them, but I have programmed. So if we take at least our 4 test groups here with the rat snakes, water snakes, pit vipers, and skinks, and these are their ranges, and I have them sampled throughout most of their range, and we ask, do they show the same patterns of diversification given area and time of origin? Well, I made a program called the Misfits, and I've got several of them, but I don't make crayons out of them, I just put them on my website, so if you want them, they're there. Because if you make crayons, then you suffer dealing with people your entire life. So basically, this takes 21 models of diversification. It actually uses coalescent diversification models as well. And this fits because they see how poorly fit your data are to any one of these models. It takes AIC weights and looks at over posterior probability distributions of trees and can ask what are your top models for these things. I also have 2 sister programs that do this with morphology. One is Minor Threat, and the other one is Bad Brain. So it's my East Coast hardcore punk rock package. You can get freely. So, but today we're just talking about diversification. So if we look at this, the highest posterior probability suggests diversity dependence for all of these. That is the early bursts of speciation that decline as carrying capacities are reached. Okay? And so I think that's a pretty neat expectation. So it does suggest that diversity dependence is happening. Now, if we look at this a little bit in more detail and we start simulating around these and simulate high extinctions, because as you should know, extinction is very difficult to estimate from biology, but can be done. And we simulate with higher levels of extinction, that's in red, we still see a pretty tight fit between what we estimate and what the simulations are saying about these organisms, okay? There's a couple of things that I want to point out that we have a very big disconnect in comparative phylogenetics, okay? And I think the phylogeography stuff here and the species delimitation stuff that we've been talking about are crucial, super crucial for understanding things like diversification. And so I built another program you can ask how bad are things, as I look at this question about how messed up you are. And so this one will say, if this is my species phylogeny, and I don't know all the cryptic species, right? They're cryptic. If I haven't done phylogeographic estimation in every one of these branches, it's possible that there's more species, right? Well, we can correct for missing species in these diversification models, except that probably most cryptic species are biased towards being on the terminals and not being deep cryptic species, right? You don't expect missing cryptic species to be this far back. So what I did is build this code that will take your long branches and put species on those long branches, okay? Based on the probability of the length of the branches, or you could base it on the length of the size of the range of the species, suggesting things with large ranges may have multiple cryptic species. And so you can keep adding these on your phylogenies and see how will this break down. And so what I want to point out is it can get pretty bad. If your gray line represents the threshold for being a birth-death model, and the more negative you go is support for density dependent or diversity dependent, that is where you diversify the carrying capacity. What ends up happening is if you have, in this case with the Lampropeltines, with about 20 additional taxa, the pattern breaks down and becomes pure birth death. And in some it's not that bad. So if you're expecting your cryptic species in your group could be somewhere between 10 and 20 additional taxa, or 30 to 50% more species that would be on those terminals, you may not be able to estimate diversification very easily from these. Now, another thing that should be obvious to the coalescent model is here, and I've written about this, but nobody cares about it because comparative guys don't deal with species trees very often, and species tree guys tend not to care about comparative phylogenetics as much. But I think there's going to be a revolution, and this is from Beerley and Edwards, 2000. And if this is the timing of speciation, this is when speciation, sorry, right here is when speciation happened, and this is when your genes diverge, right, earlier than species times, then what happens is, if you look at this over the timing of phylogeny relative, that difference between speciation and gene splitting is really high relatively early on in the phylogeny and mellows out as you get toward the older. So guess what that does to your phylogenies when you try to do diversification analyses from gene trees? It takes your young nodes and compresses them back. What's the pattern when you have all of your nodes originating early, or most of them? Early bursts of speciation that slows down. So what ends up happening is you bias towards diversity dependence. Okay, just a couple of things to go, and this graph pretty much shows that. If you generate trees under a pure birth depth model, and you estimate those trees with species tree estimators, it always gets birth depth model. Birth depth model is here at 0. Sorry, you can see that. And if you use gene trees, and these are the coalescent units, these are the hard ones to get, right? Large population sizes in small times. What happens is your gene trees will always estimate diversity dependence. And that mellows out as your coalescent units get larger. That is, you have longer time and smaller population sizes, but you don't know that. Anyhow, so just 2 points on scary things. So let's get to the community assemblage. So now we know we have our source pool, we have our similar area of origin, our timing, and our diversification. So what about these things? Species interactions, environmental filtering, and neutral processes, as we're looking at on this graph. And this is where things get pretty interesting, I think. Now, the basic ideas behind this community assessment with phylogenies, and if you're using phylogenies as surrogates for traits, which may or may not be a very good idea, and I'll show you why it may not be a good idea. If you had something changes over ecological and biogeographic space, each one of these circles represents a community, here you would see some sort of filtering where at this point you had all kinds of taxa occur, then you're getting only these guys, and finally by the end of this, over this ecological gradient, you're only sampling from this clade in your community, okay? And in this case, you have no reduction, which could be an estimate of competition, which is you have everybody from every main clade occurring throughout these communities. Now, this is very coarse, and this is the way you can see Webb's analysis in 2002 would do. There's a lot of updates. I'm going to show you some of them. Some of them are my updates. So what I did was take 122 localities. A lot of these I surveyed throughout the U.S. You can see one of the reasons I'm down here is on a goodwill mission to get some excellent Mexican collaborators so we can all work together and figure out these problems, because there's a little problem here. My communities don't occur in Mexico. But what you need, now that's not like I'm being mean about it, it's just that you need super well surveyed communities where people are surveying for years and years and years at places. Snakes are not easy always to find, so you have to survey it for years. So if you look at this map, this is where I've surveyed all of these, or some of them, and gotten some of them from the literature and press, and the size of these bubbles represent the size of the community. Now, what I did is, I think this is probably for the first largest, nearly continental sized country, I built a phylogeny of every snake in the United States, right? So this is the big phylogeny of snakes in the US. And I have traits, I have size, parity, diet, clutch, dial, pattern, and habitat for every one of these species. Now, first let's just do our basic understanding of what's going on across these communities. Well, there's massive turnover, if you can see this. So if you get around 1,000 kilometers away from one community from the other, you have nearly complete turnover, which is most of the species are not shared between those communities, okay? So we see there is a big distance function. They're really different, these distances. And if we look at something, I think this is pretty cool. if you look at longitudinal distance, you see that the communities remain pretty much the same size. So if you're in California, you look at a community of snakes, it'll be about the same species richness as you would in North Carolina at the same longitude, okay? Doesn't matter that they originated earlier here, you still have the same species richness. Now that's a key component to this. And then here's the typical latitudinal gradient of communities. This latitudinal gradient shows you that your species richness declines pretty quickly as you go into the northern regions. Now we're going to take a pretty cool metric, phylogenetic species variability, and we can, with this metric, ask phylogenetically how closely are these animals in these communities. And this will help us tell us something potentially about biogeographic processes and potentially about competition or filtering once I layer this with traits. In this example, you see the species are very closely related, so you have 0.75 as their PSV. Here they're getting more unrelated, and here they're maximally unrelated in this single community. Okay, so PSV gets much, much higher. So in this case, this might be like comparing 4 snakes. This might be like comparing a turtle, a snake, a slug, and a jellyfish, right? And so you would have maximum difference here. Okay, and just to see how that plays out, just as an example, you can go to my mom's house in Florida, and on the phylogeny of snakes, I put their pictures on here over this phylogeny, over the source pool, we can see that these are distributed roughly throughout most of the clades, and they have a PSV of 0.65. If you go up here to northern Pennsylvania, you see only this one clade of snakes are really being sampled. These are your water snakes. And the PSV is much, much lower. And you can turn this into a turnover statistic, a phylogenetic turnover statistic as well. So the questions here are, what drives these patterns, these phylogenetic species variability patterns? Knowing that species richness at the same longitude stays the same, and that it declines with latitude, what's driving this? So I use Bayesian model averaging, which is similar to a Bayesian version of a multiple regression, but assesses things like first order interactors and deal with all kinds of really cool modeling stuff, and I'll show you the results of that. And so I have predictors such as environment, temperature, precipitation, net primary productivity, and I also have biogeographic distance. And I think that's an important one because you have these things that are really far in distance, and you know they originated in the West. Perhaps PSV is predicted by just where you originate and where you end up, okay? And so when we take a look at this with the Bayesian model averaging, you can see all the predictor variables here. And this is the cumulative posterior probability, okay? And you can see that the best models, for sure, always have longitude, okay? So that's your big biogeographic distance. And your second ones have longitude and some impact of temperature. And as you get to these other ones, latitude, net primary productivity, and precipitation, they do very little, okay? I'm gonna put gap land covering this soon, but I do not have it yet. And so what we see is a very strong prediction of PSV declining with longitude. So this is weird. As you go from west to east, you don't lose the number of species. But what you do is you decline in phylogenetic species variability, such that the species in the eastern part of the United States are much more closely related to each other, even though they're diversifying to have roughly the same relative species richness, okay? And you see a little bit with temperature as well, and that's really seasonality. Now remember, I measured all of those traits, so what the heck is going on with the traits? Did we show a one-to-one? So think about this. If you're now sampling from a smaller area of the phylogeny, because you should be if your PSV is lower in the east, right? You should be sampling from a smaller trait pool too, if they're very closely related and the traits are constrained on the phylogeny. And so obviously dumb things like trait mean are associated with phylogenetic species variability, but what I think is really cool and insane, we're going to deal with this in a little bit here, is that trait variance in communities always remains about the same and high. It is not associated with phylogenetic species variability. So the types of traits regarding egg laying and diet and all this kind of stuff remain high, even though the phylogenetic species variability is going down. Well, that's actually kind of counterintuitive with what most of these community assemblers would have suggested in 2002. Now, you can also ask, is this a stable situation? This is work in progress. What I've done is, this is Herculean tasks. These are all the modern sites. These are all the Pleistocene sites for snakes, okay? And these are mapped onto the Pleistocene environments, okay? So I took these things spatially. So if this is boreal deciduous forest, it resembles, at least in shape and area, where the modern deciduous forests are, okay? And same things with grasses and deserts, okay? So I grouped these modern and Pleistocene by their ecoregions. And what I wanted to ask is, phylogenetic turnover more extreme through time, from the Pleistocene to the Holocene, or more extreme through adjacent communities, right, in their own time frame. And so what we find here, and this isn't completely done, is that through time, and this is pretty extreme, they're very similar. So if something's, and there are no extinct snakes in the Pleistocene, believe it or not, right? So they're all, so the biology that I'm using for modern things can be used for the Pleistocene animals. And so what we see is, if you're going to have replacement in the same ecoregion, it's always going to be replacement of something from that same clade in that ecoregion, such that your phylogenetic species variability is extremely, or the turnover is extremely the same through time, but across regions always remains really, really high, okay? So I would suggest that at least in the United States, this is a very stable system. Now, if we look at local assembly, we can use these regional biogeographic components. We already know it's there. We know longitude is some predictor of phylogenetic species variability and some aspect of trait variation. And trait and PSV are disconnected. So what the heck is going on here? How do we assess traits and phylogeny at the same time? So you could have phylogenetic clustering with very different traits, which is what this looks like. You have things that are very closely related with very different traits that allow them to be in the same area. Or you could have phylogenetic overdispersion, where you have very different organisms, but they all have the same kinds of traits. And so this is not known from the phylogeny alone. So what we got to do is make up some cool metrics, right? And this is from Cadet et al. And I've modified this a little bit, so if you want the code, it's much easier to use now. And so you can take your phylogenetic distances, okay, and you can take your trait distances, okay? So you have all of your trait distances, and you can put them into a single metric by community. And it can be controlled by this tuning parameter. So here's your phylogenetic distance. Here's, they call it functional distance. Functional is trait distance. And this is your tuning parameter A. So if A is all the way to 1, then you see this functional phylogenetic distance is completely controlled by phylogeny. If A is close to 0, then it's completely controlled by your traits. So this is reconciling the variability in communities between traits and phylogeny. Okay, really quickly here, we can ask, are these diverged or converged communities? So remember, this is your phylogenetic distance here, this is your functional distance, and this is the combined measure of this. So if A is low and your functional phylogenetic distance is high, that means most of your community are being driven by trait differences. As you slide A towards 1, and it's high, that means all of your differences in your communities are phylogenetic differences, okay? And that could be that you're just missing the traits that are variable there. That's possible. There may be other physiological traits and you haven't seen them. So in this case, you would have a very divergent pattern. You have species that are closely related, but their morphologies are very different. And in this case, you would have a converged pattern where phylogenetically the species are very different and the traits are the same. So when we look at this over all 123 communities, we see massive trait variance relative to phylogenetic variance in those communities. So they're very, very different. And it's a divergent pattern. And we can use, oh, by the way, these are Bayesian correlation tests. So if you want to do Bayesian correlation, there's this thing called Bayesian Band-Aid, which is really damn cool. And it downweights because Bayesian downweights weird outliers and things like that. And that's in R as well. So that's where these kinds of graphs come from. And you can do the Bayesian model averaging, and you'll predict that things like temperature and precipitation now are much bigger drivers of communities when you take into account what's driving the communities in terms of morphology versus phylogeny, okay? So major differences in morphology and very few differences in phylogeny relative to that morphology. So what you're seeing is, just like when I showed you the variance was really high over PSV, the creation or the maintenance of very different traits to deal with these environments or deal with things like competition, even though the phylogeny is saying they're closely related, and that doesn't So the phylogeny alone, this is a key point, is not helpful in community analysis. So now if we look at traits in community, we haven't really answered the question, what the hell is driving this? I've just told you the patterns. This is just kind of the last part. Is it environmental filtering? So again, do you have traits required for colonization? And are these traits yielding convergence? And again, you can ask, you can have ecological similarity that again limits these coexistence. And we can test this with these Durate and Pilar models that are also in R as well. And so you can integrate traits, environment, community, and phylogeny. And again, just to go over this really quickly here, if you have an ecological gradient, let's say from warm to cold, if you maintain maximum differences in your communities, people would suggest that is competition, right? They're maximally different. If you see that you're filtering from one type to the other as you go from cold or warm to cold, whichever way I said, and you're only getting certain clades, then that is has used to be called habitat filtering. And then in this case, if it's just totally random, it's a neutral process. But just quickly, there's a huge problem here, guys. It's okay, it works fine if the traits are always associated with the phylogeny, right? So that if you're picking 1, 2, 3, and 4 out, and they all have this trait to deal with warm, and the green guys all have this trait to deal with cooler, and the blue guys have all this trait to deal with cold. But if you have this situation, it sucks, right? Because if you were doing this with phylogeny by itself, you would say, well, you have no pattern here, because there's your red one, there's your red one, there's your red one, there's your green one. They're not in the same place. So the traits have re-evolved to, potentially match these communities, right? So you have to know something about your traits. Now, what I've done is taken and done some, I think, pretty neat stuff. Take that phylogeny, multiply that into the community. So now we have our community phylogenetic estimation, right? And so that's our community matrix. We take all our traits and multiply those into the communities and use something called fuzzy weighting, so we can see the contribution of different kinds of traits in the area, and the traits that are maximally different, so we can see if there's divergence among the traits. And as well, we multiply the different kinds of ecologies into these communities. And from these, we can do a series of Mantel tests. I'm going to update this for better modeling using phylogenetic least squares modeling or something, because I'm not super trustful of Mantel correlations. But if you do this, you correlate phylogeny with traits. So are the traits correlated with the phylogeny in the community? Are the traits correlated themselves with the ecology in the community? And then finally, is the ecology associated with the phylogeny? Okay? These should be striking, well, they are already, but I'm just saying. So these should be striking results, but maybe not. When we look at this just straight away, we can see our big generator with increased seasonality is increased live birth, right? That should make sense to you. If you're going to live in really cold environments and go from warm to cold really rapidly, probably the worst thing you can do as a snake is lay eggs. Why is that? Huh? They die, exactly. So what you see is, and you see a lot of weirdly darkening of animals in the north, and the females will move around with her babies inside her and keep them warm, right? And so don't lay eggs. And so you see major habitat filtering with increased seasonality. You also see an increase in endothermic diets, okay? Eating mammals and birds, okay? And that's high energy stuff. So if we look at this, we can see our habitat filtering is pretty important for parity, having live birth, and diet, a little bit for ectothermic. So you see this trait divergence. If you're going to eat ectotherms as you go into different environments over these gradients, you're going to eat different kinds of ectotherms. They don't overlap, okay? And so size and habitat, being arboreal, et cetera, have no structure to them. So there's no phylogenetically structured assembly patterns with respect to traits or environment over these gradients. So the phylogeny does not predict who would ever be there. And it's not correlated at that level with the traits or the environment, even though the traits, using a Longberg's K over the entire phylogeny, would be correlated and show phylogenetic signal, but not at the community level, okay? And so this goes with some of the work that Alex Pyron and I have published on bioparity and evolution in small mates. This was just out in Ecology Letters. And we used a phylogeny of 4,200 small mates and asked, if they're viviparous or oviparous, what is actually happening. And what we see is several changes back and forth from egg laying to live bearing to egg laying. That may not seem extreme. These are freaking embryos. How many birds have live birth? 0. How many mammals lay eggs? 3, right? But these guys jump all over the place. And they do this, cool enough, guess what? When it gets colder, it's much better to have live birth, evolutionarily. What's actually also cool, using those same models of diversification that are implemented in BISI, we can see that speciation rates are actually higher in viviparous animals than they are in egg layers. But wait, you guys should go, wait a second, you said there are more viviparous in temperate regions, but diversity of species are much lower in the temperate regions. Well, guess what? Extinction is much higher also in the temperate regions. So you may be producing a lot of species, but they're going extinct rapidly. And that's another explanation for these latitudinal gradients, right? It's a harsher environment, harsh to deal with. Uh-oh, my nervous man is getting up. I'm worried about it now. Okay, so I'm almost done. So what makes a community? Ancient biogeographic history, right? So dispersal from the old world to the new world. This is giving us our source code. And we see these regional biogeographic history. We see this diversity dependence. So if you're landing in the same spot at the same time, you see this diversity dependence of diversification, and ultimately a dispersal from west to east. This should show you another reason why snakes are good models. They have these very predictive patterns of which you can test direction. And you see this declining phylogenetic species variability in spite of stable species richness in this dispersal from the west to east, okay? And if you look at these trait environmental interactions, we see filtering for traits, but we see no trait community phylogenetic structures completely absent. So trait variance is maintained in spite of phylogeny, likely filtered by these areas. So what I would say is the way forward with this stuff, and you know, in 2 years these things may be correct, right, with better models. So one thing that you can have, and I know you have this, is if you look at this, look at species 1 here, right? It's possible that in different environments, the same species changes their traits. Oh, that sucks, right? But it's actually cool. So because you may actually have a better signal for filtering. So here, species 1 is doing whatever trait 1 is here, then green, and then blue. And this, we know this happens. Things like Actisteron and water moccasins. If they live in heron rookeries where there's lots of birds around, guess what they eat? They eat birds. When they live in areas where there's lots of fishes around, they eat fish, right? So there is some changing of these traits through environments that need to be accounted for. And then, as well, getting all the next-gen data so our trees aren't goofed up, right? We want to have good trees, and they're probably still goofed up, but they're maybe less goofed up kind of dumb things. We want to find all those cryptic species, so that's why I have a grant in. I think we need to, as I've been saying, we need to join the US and Mexico together to find all of our cryptic species, and 60% of our taxa here share this, go across the border. So we really need that. We need our species trees. We need to integrate all of these models a little bit better, the models of biogeography, diversification, etc. We need to integrate those. We need the tropical communities. We have to have the tropical communities. And my mind is blown by the great interchange between South America and North America and what's going on in those communities. Because you have diversification in 2 different continents meeting. So what's going on with filtering and habitats in there? And then there seems to be no link between diversity dependence and competition. I found no signal for competition, even though diversity dependence, you're producing more species. So there's a disconnect, as I was saying at the beginning, of some sort of scale disconnect here, where you see diversification slowing down, but you don't see at the community level any kind of competitive interactions that would slow speciation, perhaps at the regional level. And we're missing out. But that's the end for now. But wait, you know, I got some good credits. There's a lot of my lab nerds that were super helpful and the greatest people on the planet Earth. Alex is no longer in my lab, I'll just always say he is. And tissue providers and coding advisors, and clearly funding is super hard, you know, these days. And, you know, finding funding is difficult. I always put crossword puzzles in my evolution tests for free credit, but they're definitely there. You can find them. They're around. So thank you guys very much for listening to all this snake stuff.
Muchas gracias, Dr. Goldring. No sé si alguien tiene preguntas.
La voy a hacer en español.
If you want an answer.
I'm intrigued by the results, and I'm wondering whether, on average, communities out of northern latitudes are older on average than the ones in the west, let's say the southwestern states, towards the eastern states.
The communities are definitely, at least the diversification and the level of communities are older in the north, and we tested this with other things. As you go towards Central and South America from the north, the species are definitely younger in that direction and in the direction of the east, right? But I don't want to say that 100%. I don't have the phylogenies for all of these guys. The colubrians are extremely difficult to get, because what you could have is, even though they originated in the West, what you could have is extinction, recolonization, and all this stuff, and the original signal is gone, right? I know in the Southwest, as we approach the Mexican border, and likely in Mexico, I have some communities in Panama, the phylogenetic species variability is out of control. It's massive, right? So you have very divergent things in communities that are coming up in the Southwest. So that's another piece that's elevating the phylogenetic species variability in the West relative to the eastern ecoregions of the US, right? Not only are those younger, they don't have contributions from these really strange or divergent lineages.
And so by then understood, the communities on the east are more composed of closely related lineages,
Yes,
right?
yes. Only 3 deep lineages that are very closely related, absolutely. And they're mostly things like, if you know what Neroidian family is, so water snakes and garter snakes. Lots of very closely related water snakes and garter snakes, and lots of very closely related rat snakes. But we don't have lots of things like, we do have, but not lots of them, like Tanphila, and all of these strange things you see in the Southwest, like where he would see all of his, you get weird communities of older things, right? But the niches are still being completely filled by these younger things, if that makes sense. And the richness is the same. But we really need Central America. But as I said, once you get in communities that are 60 species, it's much harder, because the worst thing you want to do here is say something's not present when it absolutely is present. And we really want frequency as well, which I don't have. Frequency adds another, frequency of who's common and who's not common adds another statistical, very cool element to this, because if something's really common in the absence of something else, and then when they're together, it becomes very less common, and you see the effect of competition over communities. And I have this for lizards, and that's kind of the next product. Reduced to working on lizards. More questions? You have to have questions. You can ask me. You can ask me about my shoes. But I don't care. You know, if we go out here, I don't care. Your English is not so good. My Spanish is terrible. It's lots of dirty words, and then mixed up with Portuguese words, and it sounds like a very mentally ill child is what I sound like. So don't worry. So if you want to ask me something in English, we'll figure it out. It'll probably be more fun that way too. So.
Esta sesión, pues esperamos a las 3:20 para la mesa redonda y los comentarios finales, y después la premiación del concurso de fotografía y la clausura.
So,